Good evening to all lecturers and audience. My name is NURUL Ayuni binti Ahmad. I am student from Bachelor of Science in Aquaculture. Today I would like to present my current research entitled Xestospongia Associated Microbial Community in Different Locations. What is marine sponge? Marine sponge is efficient filter feeders. They have primary and secondary metabolite and bioactive properties inside the sponges. Hence, sponge-associated microorganisms promising as pharmaceuticals. They produce novel antibacteria as sponge associated with marine bacteria. Then, sponge also key element of biogeochemical cycle in coral reef. The relationship between marine sponge and some of their bacterial and archaea symbionts. The bacteria symbionts may play roles in digestion, waste removal, and chemical defense for the sponge host. Then, with the involving of antimicrobial activities, the archaea member of sponge symbiotic microbial communities may play an important role in nitrogen cycle of the ocean. The problem statement is there is a scarce paper, reports, journals on Xestospongia related bacterial and archaea community in Malaysia. Then there is a little information exists about the taxonomic affiliation of sponge symbiotic microorganisms, especially archaea. The significance of study is to revealing microbial community in Xestospongia might provide geological difference in the same sponge species. The objective is to compare bacterial and archaea community with Xestospongia among 3 islands, which are Bidung Island, Perhentian Island, and Kapas Island, using denaturing gradient gel electrophoresis, which is DGGE. The study sites are Pulau Bidung, Pulau Perhentian, and Pulau Kapas. The steps from sample collection and processing are sample collect, then DNA extraction by using step method, then PCR using GC clamp primer to identify Xestospongia using specific primer for bacterial and archaea, then denaturing gradient gel electrophoresis, which is DGGE. Then from band present in DGGE, we will run for alpha and beta diversity microbial community composition. Then from the DGE also, we will run for band exercise, run PCR, sequence, and phylogenetic analysis. For the sample preparation, we will wash sponge with sterile artificial water 3 times to get rid of loose starch, debris, and sediment. Then we will cut the sponge. The sponge will preserve by immerse them in DNA buffer. Then sponge will storage in -80 degrees Celsius. For DNA extraction, we will use step method. Step method is traditional method during extraction of highly polymerized DNA from plant material. Then we will run for the natural gradient gel electrophoresis, which is DGGE. DGGE is a method by which fragment of partial 16S rDNA, amplified fragment of identical length, but the different sequence can be resolved electrophoretically because of their different melting behavior in a gel system containing a gradient of denaturation. The expected result is it is expected that there is different microbial, which are bacterial and archaea community associated with Xestospongia among 3 different islands. So this is my Gantt chart and references. That's all from me. Thank you.